everyone. I believe that, hello. I will start presenting myself and the speakers of this session, so of this bioprotocol webinar. Meanwhile, I think you are coming in and you can introduce yourself using the chat. Yeah. I'm Irama Cordeiro. I'm a full professor at the Federal University of Rio de Janeiro, Brazil, working in protein biophysics. And like in the past 7 years, we are really into studying biomolecular or condensates dynamics and in general. So today, the lecture will be held by both PhDs, so Dr. Mariana Giuliani do Amaral, she's an assistant professor here at the UFRJ, as well as the Faculty of Pharmacy. Mariana do Amaral is one of the pioneers in studying biomolecular condensates in Brazil, I would say in Latin America. And so you can check later on her CV. And after we'll have also a presentation by Dr. Aline Passos. Aline Passos is a research scientist at the Brazilian Center for Research in Energy and Materials, CNPM, and she works in the Brazilian Synchrotron Light Laboratory, which is our fourth generator serious synchrotron light source in Brazil. So it's high and top end techniques that she's specialist. And today she'll talk about XPCS applied to study the condensate dynamics. And Mariana will talk about fluorescence recovery after photobleaching assays. So both of them are expert in these techniques. And I hope you enjoy this webinar. And after all, I'll be here taking notes for the Q&A after the presentations. Welcome, everyone. Thank you. So I guess we have already 57 participants. Hello. So people from, I will have just meanwhile, there are people from China, Czech Republic, US, France, India, and lots of Iran, France again, Italy, Spain, Israel, Singapore, lots of different countries. So it's a pleasure to have such an international audience with us today. Yeah. Well, and Luis Felipe from Brazil. Hi, Luis Felipe. Nice to see you here. So I guess to make the time our schedule okay, I think we can begin. Mariana, do you want to also introduce yourself and begin with your webinar?
Yes.
Thank you.
So thank you very much. Yeah. Thank you, Iraima, for the kind introduction. Iraima was my PhD advisor, and she's also an expert in this team. She supported me unconditionally in learning these techniques. And I'm very happy today to introduce you to some of the techniques we explore in this bioprotocol. I think our protocol is unique in the sense that it's a very multi-parametric protocol where we use multiple techniques to characterize condensates. And I also thank Renee and Antara for this invitation. I'm a big fan of Bioprotocol. I think it's a very important journal for the field because the protocols are very detailed, are throughoutly reviewed. So every time I want to learn a new protocol, I go to Bioprotocol. So thank you for this very nice journal. So starting with a very brief introduction about phase separation. Well, I cannot change my slide somehow. Okay. Okay, there you go. So our agenda involves talking about how these condensates can be linked to neurodegenerative disease development, because it's known that these condensates, they are formed by intrinsically disordered proteins that can transit from a dynamic state to an irreversible solid state. Also, we will explore how can we characterize both liquid and also other material states of condensates by microscopy, and we can make use of extrinsic probes for that, for example, Cypro Orange. We also will explain a little bit of fluorescence recovery after photobleaching and how to analyze and process this data to probe these phase-separated states. And finally, Aline will join us to explain about XPCS and how this synchrotron technique can also add in the characterization of non-equilibrium phase-separated systems, okay? So, what are biomolecular condensates? So, peculiar proteins with prion-like domains or regions with low complexity, they can self-assemble in a dense phase that is completely devoid of any lipid components. And this dense phase that can harbor very high protein concentration is in continuous exchange with the light phase. So turns out these intrinsically disordered regions are mainly responsible for driving these condensates. Usually these proteins contain long regions, but also some oligomerization domains like coil-coil can also drive phase separation. So, a pioneering work by Brandywine and collaborators in 2019 described this phenomenon for the first time, where we see P granules that are canonical non-membranous organelles, sorry. They undergo this dripping. These are nucleus, so they can be very dynamic at this perinuclear region. So other groups over the 20 years, the last 20 years, also characterized this phenomenon that is very transient and reversible. So here we see G3BP1, that is a stress granules protein expressed fused to GFP. And upon stress, for example, cold shock or oxidative stress, these granules can form. And these can be reminiscent of very fun lava lamps, where we see these fusion, fission, and deformability, which are liquid-like hallmarks. So numerous membranous organelles exist as liquid condensates, both in bacteria and eukaryotic cells. And we already knew that these were membreless compartments for a long time, like Cajal bodies and even like P bodies, but we didn't know the mechanism, and the mechanism is liquid-liquid phase separation. But in addition of all these membreless organelles, there are transient proteinaceous biomolecular condensates that can also form, and they can account for a myriad of functions. So they can have a size that is like 0.2 microns to 10 microns in the cytosol of animal cells, and they can be responsible for many processes. For example, condensates can hitchhike lysosomes and aid in the transportation of granules throughout microtubules. Also, we can have condensates at the cell surface that are very important for cell signaling. And usually these condensates can also contain RNA and be important for transcriptional control as well as RNA metabolism. So many condensate forming proteins that are these ones that we studied for a long time, for example, FUS, TDP43, alpha-synuclein, and tau, they have this flexible dynamic nature, and they are involved in neurodegeneration. So it's being well characterized that these proteins that form these intracellular and neuropil aggregates for all these different diseases, for example, Parkinson's disease, Alzheimer's, and so forth, they can undergo phase separation when overexpressed in live cells, and we can reproduce that, reconstituting the components in vitro and dissect really the material properties of these condensates. So why is that important? Because these condensates can have a very high protein concentration, and they can constitute a non-canonical misfolding pathway. So if these liquid droplets undergo maturation or hardening, they can form more gel-like states that are less dynamics and evolve to amyloid fibrils formation. So it's very important to study phase separation mechanisms. And this has been shown, for example, in the case of tau, phosphorylated tau, that aggregates can emerge from the droplet surface. And many other groups have described liquid to solid phase transitions associated to aberrant phase separation. So our group studies the prion protein that is involved in fatal brain diseases as well. But what we didn't know were like the modulators of PRP phase separation. So there was a pioneering work that described in vitro PRP, or the prion protein condensates, but this study used a very high protein concentration, like 50 micromolar, that may not correspond to the intracellular concentration of PRP. And we wanted to know the effects of copper ions in the modulation of the prion protein. Why is that? Because this is a very well-known ligand of PRP. It has been studied for more than 25 years, the effects of copper in PRP interaction and also aggregation properties. Also, we wanted to know if these PRP condensates really exist in cells. So PRP has half of the primary sequence is intrinsically disordered, and half of it is a globular domain enriched in alpha helix. And in this N-terminal domain, there are histidines embedded in octapeptide repeats, and these can coordinate copper ions. So PRP can bind up to 6 copper ions in the N-terminal domain, and there are other histidines in the C-terminal domain. So PRP is also a GPI-anchored protein that is found at the plasma membrane. And it has been characterized that it can function as a metal ion sensor. Storage can also be important for oxidative protection, for example. All that because of this very highly propensity to bind copper ions. But we didn't know if copper ions can really trigger the formation of the prion protein condensates. So to investigate that, we use the techniques that we described in this bioprotocol. We use in vitro and live cell approach. And we thought about using a more physiological-like relevant buffer, in which we searched about the intracellular concentrations of ions, and the concentrations are here, and you can find in the protocol. And also we use a molecular crowding agent like PEG-4K or 20K to mimic the crowding of the cellular environment. We also based our assays using relevant concentrations for copper ions that are, for example, found in the synaptic cleft. So first of all, we need to express the prion protein and purify that to conduct the in vitro assays. So we use a plasmid where we modify this plasmid to do not contain a his tag, because we wanted to only evaluate phase separation in a tagless protein. This is very important because these tags can modify phase behavior. So we firstly transformed this PRP plasmid in E. coli BL21. Then PRP formed these inclusion bodies that are amyloid-like structures. And if you do TEM of these inclusion bodies, you can even see these fibrils. So for that reason, we have to use denaturation reagents like urea or guanidine to really dissolve these aggregates. Because they are held by very strong hydrophobic interactions. So after we use these denaturing agents, we inject the protein in a his trap column that has nickel immobilized. So we use the intrinsic ability of PRP to bind to metal ions to also do its purification. So as I showed before, PRP has these histidines in the N-terminal, and it can also coordinate nickel that is present in the column. So after we bind the protein, we have to do a refolding. So PRP can adopt its native folding. And we do a quality control of that. We run circular dichroism to check if it adopted like the alpha helical C-terminal domain. We also confirm that the disulfide bridge was formed. So we use imidazole to elute the protein from the column. And after that, we carry out dialysis to exchange to water, for example. So it turns out PRP can be stable in water. So this really facilitates us to use different buffers. So of course, you have to do protein quantification afterwards. And I recommend you, if you work with aggregation-prone proteins, that every time you thaw an aliquot, you have to adjust the concentration. You have to check the concentration once again, because these proteins are very sticky and they can form small aggregates. So you have to centrifuge and remeasure the concentration. So how to investigate condensate formation? So we can investigate by a very nice microscopy that is differential interference contrast, FRAP. And FRAP is very important to access these different material states. So starting with DIC. For DIC, we don't even need the labeled protein, right? Can be very nice because some dyes can also impact phase separation properties. So for DIC, you firstly have to check if you have the appropriate objective for DIC and all the necessary components. For example, the normalizing prisms, in the polarizer and analyzer. So after you check all the DIC components are in the microscope, you have to adjust the illumination by the method of Cooler. And we use these very nice imaging chambers that we assemble using double-sided stick tapes, forming like a sandwich here to visualize these condensates. So DIC enables us to have a very good contrast of these transparent circular structures that are the condensates. And we have this shadow casting appearance here, like a pseudo 3D. So we can also probe the protein conformation in condensates. And for that, we propose to use the cyper-orange dye that can be excited near the DAPI channel, the 488 channel, the GFP channel. And this dye is normally used in DSF assays, where it's known that when the protein starts to denaturate, it can interact with cyper-orange. And also amyloid structures, they can interact with this dye. And also it can have a 3 times more increased emission than TAT. So cyper-orange is more sensitive for detecting amyloid structures. So we use a laser scanning confocal fluorescence microscope to probe whether these condensates can bind cyperorange. And because cyperorange comes with DMSO, it's necessary to do a stereo dilution. So you reduce the concentration of DMSO. So to avoid any artifacts that it may cause to the aggregates, condensates and aggregates. But of course, please run controls only with DMSO, and we have done that. But for this presentation, I'm showing only the sample that we imaged and has a very high cyperorange fluorescence in these condensates that we know are more gel-like and solid-like because we probe with FRAP. We also did cyperorange staining of the C-terminal domain of PRP in a very high concentration, and it can even depict like some heterogeneity inside the condensates. So FRAP is not a new technique. It's been like 50 years since the first inception of FRAP. It's been used to study mobility of proteins in cells, and it's very informative because you can extract quantitative data. So for example, in this case, an ER protein was bleached and it was completely recovered at a high temperature, but then you can modulate the temperature or the conditions, and you find that this ER protein cannot recover the fluorescence anymore after the photobleaching. And this can be very useful for describing condensates. So in the case of condensates, it's very important that if you want to probe the diffusivity and dynamics of the dense phase, that you bleach only intradroplet regions. So this can be challenging, but it's important that you don't bleach the whole condensate. In vitro, you can easily draw an ROI intradroplets, but in cells, this might be very challenging, okay? So it's important for studies of the material states of condensates. So we wanted to study whether copper ions could modify the mobility of PRP. And I mentioned before that this protein is anchored to the plasma membrane. So we wanted to bleach PRP in the presence of or absence of copper ions at the cell surface, and we saw a very interesting phenotype that when we incubate cells for 1 hour with copper chloride and these cells express PRP fused to YFP, we see an accumulation at the cell-cell interfaces. So we wanted to compare the mobility of PRP in the cell-cell interface versus individual membranes that face the extracellular milieu. So this was very important for us, a very important collaboration with Professor Susan Wegman and a very talented and brilliant PhD student, Satabdi Mohapatra, that really helped me in a research stay where I spent 7 months in DZNE characterizing this phenomenon. So first of all, we transfected HEC293 cells because these are very easily manipulable cells. And after 2 days, we run the treatment with copper chloride that was until 1 hour. After that, meanwhile, we already turn on the stage in the microscope so it can be pre-equilibrated before the experiments. And we use a spinning disc confocal for that. Also, it's very important that you turn on the perfect focus system. That is a focus system developed by Nikon. That is an autofocus that will avoid any drifting during FRAP excision because FRAP is basically a time lapse, right? So we use a 60 oil immersion objective, and we adjust the laser power accordingly. So you have to do that for your protein of interest. Of course, less laser power, the better, so you avoid phototoxicity. That's why it's also very nice to use a spinning disc. and we use 200 milliseconds exposure, okay? Then we looked for cells that showed moderate expression levels of PRP or IFP, and we drilled the ROIs of the same size in each of these membranes. So this is very important, that you have consistency in the ROI size. So basically, we copy and paste exactly the same diameter because this will impact in the FRAP analysis. And of course, draw only one ROI per membrane. So we set the bleaching protocol beforehand and we tested that 100% laser power for 400 milliseconds was enough to bleach in an extent of 50 to 70% the initial fluorescence. And of course, I do not recommend UV photobleaching with 405 in the case of cells because it can be really cytotoxic. And after the bleaching, we monitor the recovery for 2 minutes, but you have to monitor until the plateau is reached, of course. So 2 minutes was enough. So this is a representative image showing the pre-bleaching and post-bleaching frames. And I will show you a time lapse. So you have to collect FRAP experiments, of course, in other fields of views and wells, and run at least 3 independent biological replicates with at least 30 time lapses per condition. At least at the cell surface, this gave a statistically significant number for us. And of course, save the FRAP time lapses and the X and Y coordinates of each ROI. Some microscope you have to save separately, so be mindful. So this is a time lapse of a FRAP experiment. This is the ROI corresponding to the background, the reference of a cell-cell interface. This is the bleached ROI. This ROI, we had to discard this data because a protrusion of the cell was evident. And this was a FRAP of isolated membrane. So you can see here, for example, here there is this protrusion and the cell moved a lot. So we had to discard this data. While we could use this data and also the isolated plasma membrane. So moving for the data processing, as we are running out of time, because Aline will also talk about XPCS. You can also read in the paper, but there is the quality control of every ROI. So it takes a while to process the data because some ROIs also show cells will move a lot and they can show unstable pre-bleaching fluorescence. So these you cannot use. Of course, you can select the reference and background afterwards because the data will be there. And actually, it's good that you have references matching the same fluorescence intensity as your ROIs. And of course, do not take a reference ROI from the same cell that has been bleached. And all these golden rules are written in the protocol. So after you extract the fluorescence values, you can use FIDI or ImageJ. We copy this data to Microsoft Excel, where we can calculate and do the background subtraction. So both the reference and the bleached ROIs, we subtract from the background. We correct for natural photobleaching. This is very important because during acquisition, photobleaching will occur. This is a property of fluorophores. And photo-induced chemical activation will occur naturally over time with the acquisition. So we also determine the mean pre-bleach fluorescence, averaging over the 5 scans we collected before bleaching. And we normalize the corrective fluorescence intensity with this mean max normalization. That is a very common formula we use to normalize data, okay? So analyze at least 20 ROIs. Of course, a lot of data will be discarded using live cells. And of course, plot the mean and error and calculate the half-time half-life time, and also the mobile phase. So we did that. This is illustrative data showing for the cell-cell interface, showing that PRP is not as mobile as the control YFP GPI at the cell surface. And with copper, this further decreases. And there is also a difference in the individual cell membranes, where PRP also has a decreased mobility, but not in the same extent as in the cell-cell interface. So copper increases densely packed PRP molecules at the cell-cell interface. So to do FRAP in vitro, it's necessary to label the recombinant protein, right? So we thought about fusing PRP to GFP, but this would not be an ideal approach, as GFP even has a higher molecular mass than PRP. So instead, we conjugated PRP to Alexa fluor 647 because this dye will conjugate to primary amines, and PRP has 10 lysines. And after the coupling reaction, we obtained a labeled PRP in which most of the molecules had either one conjugated dye or two, based on the stoichiometry we assessed afterwards. So this wouldn't really impact, we at least predicted that this wouldn't impact phase behavior. But of course, we run the experiments to check that, and this was not the case. I mean, the Alexa didn't impact phase behavior, but you have to always compare the unlabeled protein and the labeled protein and compare the phase separation properties. And this shows PRP label here by fluorescence microscopy and DIC. So to run in vitro FRAP, it's essential that you use this imaging dish with saturating humidity. So you basically add a droplet of your phase separated solution. And during the acquisition, you have to make sure it will not evaporate. That's why we use this special device. Because it's really a tiny droplet of 3 microliters we use for the FRAP experiments. And this is a representative raw data from Zeiss confocal microscope, in which you can see here in the real time the bleated ROI as compared to the reference and the background. And you will see the characteristic recovered of liquid-like droplets. And after you process with the background correction and normalization, you have this very nice graph. So now changing gears to Aline. Aline, you can take over. I will provide you control now. Yes.
Thank you, Mariana. There are a lot of questions already. A lot of questions. Okay, so at the end, thank you. And thank you, Aline.
Thank you. So in this last part of presentation, I will talk about X-ray photon relay spectroscopy, XPCS. But just before going to the protocol, I'd like to give you a quick introduction about the technique. Mariana, actually, I cannot control the presentation. Can you move to next, please?
Yeah, it says you can control here, Lynn. Maybe try again.
Yeah, now it's working. Sorry about that. So XPCS is a technique that allows us to study the viscoelastic properties and the relaxation behavior of protein condensate. So similar to FRAP, we can also have this information about the state of these condensates. And we can also study different dynamics processes that are really relevant for biological function, for example, diffusion, aggregation, phase separation, and so on. And well, I imagine most of you are used to DLS. XPCS, it's very similar to DLS, But when we use X-rays instead of visible light, we have some advantages. So we are not limited to transparent samples. So we do not have to dilute our samples. We can use concentrated samples, opaque colored samples, and we are not limited only for liquids as well. We can study like films, gels, different states. But the idea, the physics behind it's really similar to DLS. But in XPCS, we use X-rays, so we need coherent X-rays to perform this technique. And in the new synchrotron, so in the third and fourth generation, we have a high coherent flux. And when you have coherent X-rays, so we have the same wavelength, the same phase, and we illuminate a sample, we will create a speckle pattern. It seems like random, but actually it's related to the exact spatial arrangement of your sample. So then you can imagine if you measure one speckle pattern and you have information about the exact arrangement of the sample, and if things are moving on the sample, and we collected several speckle pattern, then we can follow these dynamics. This is the basic idea of XPCS, to get information about dynamic based on the fluctuation of the intensity of these speckles. So now going to the protocol. So first thing is what is necessary to perform XPCS experiment. So as we need coherent X-ray, it's a technique that we can only find in these big facilities. So we have XPCS on third and fourth generation synchrotrons, and also in X-ray free electron lasers facilities, XL. We also need a fast X-ray detector, and sure, it will be depends on the time scale of your dynamics. It's a really fast process, so have to have really fast X-ray detector. And also it's very important to have this analysis pipeline that we can see the data at the same time we are measuring, because then we can take decisions based on the final data. And just for those that are not used before with synchrotrons beamline, so I will present this small video that shows what's the beamline structure, just to give you an idea. So this was taken at Caterete beamline. So here we have this really big vacuum chamber where the detector is located inside. And here we can inject the protein solution in the sample holder, where you can control temperature as well. And then we illuminate the sample with coherent X-rays, and we will collect the speckle pattern in the detector. And when we are doing the measurement, what you see on the screen of our detector is this fluctuation of the speckles. It seems really random, but actually it's related with dynamics in your sample. Then how we obtain the data from these speckles. So we select a region of interest, and then we have the intensity of this speckle pattern over time, and we apply the correlation function. And the main result that we have in XPCS is this correlation curve that is exponential decay. So now going more specific for how we measured XPCS of these PRP proteins. So first thing, prior to data collection, it's to set in the beamline. So we have to choose the energy of X-rays that you're gonna use, the sample to detector distance. Then first of all, we measure direct beam to determine the coordination of the center of the beam. And we will measure a background, like an empty image, just to determine what regions we will mask. Then they will be outlined from the autocorrelation calculation. For example, here we'll have this region where I have these gaps between the chips of the detector, scattering from slits, and then we remove all the sample that are not, all the information that are not coming from the sample, and then you can have the correlation. So for collected XPCS data, we prepared the PRP solution. Part of the solution was loaded in this capillary flow cell. So we used quartz capillary with 1.5 millimeter diameter. The diameter will depend on the energy of X-rays you are using. And part of the sample we collected to perform microscopy in the same sample before XPCS. This is very important because then we can be sure that you had the formation of condensates. So part of the sample did microscopy and part we inject in the sample. So this sample holder was located here on the beam line. And then we have all connected in vacuum. And then you have the incoming X-rays that goes through this capillary with your sample, and then the scattered beam goes direct to the vacuum chamber where the detector is located inside, and then we can acquire the data. So before start with the samples, actually we have first sample for test because you have to determine the conditions of the experiment. And one very important thing when we are working with X-rays and proteins is radiation damage, because these samples are really sensitive for X-rays. And now in these new facilities, the flux is really high, so have to be careful about it. So first thing we did was collect the XPCS data. And then from this data, we can integrate it and calculate the Sachs curve. Looking at the Sachs curve, we can see if you have some structure changing. And it's not expected to have any structure changing in the condensates when you're measuring their dynamics. So if so, it's induced by the beam. So then what we can do is, for example, use some attenuator to decrease the dose. So this is important for choose the conditions of your sample. Also, proteins, they do not scatter a lot, so sometimes we have to do several acquisitions and then you can average the result to get good statistics. Once you find the damage threshold, the second important information is to find this dynamic time window. How long you have to measure XPCS to get this complete decay. So to choose this, we will change like the exposure time, the total number of images. In XPCS, usually we acquire a lot of images. It's like thousands of images to get one curve. And once you find what's the dynamic time window, and if it's in agreement with this damage threshold, then you can go ahead. If your dynamics it's too slow, maybe, and then we have to come back again and recheck the radiation damage threshold, and then you define the conditions of your measurements. Once you have all these conditions set, now we can load a fresh sample and then start the XPCS measurements, collecting multiple scattering patterns from your dataset. Once you collect the acquisition of XPCS data, usually it's really fast. So we inject the sample, we collect all the frames, and then what we did, we recover the protein sample using a new syringe, and then we load it again in a new microtube, because we will analyze again with microscopy after XPCS measurement as well. This is a double check to be sure that the sample we had the condensates before the measurement, and after measurements, we didn't have any induced change from X-rays on the sample. So we always did like this just before and after the measurements. And when you finish, important step, it's cleaning the sample cell, because it's this capillary, and then we use the same cell to measure a lot of samples, so we have to be sure that it's cleaning. So first thing, we use water, then we use a solution of salt. As Mariana mentioned, these condensates, they are really sticky, so they have the tendency to stick in the capillary wall. So we use the salt that helps to cleaning the droplets, the condensate droplets. Then we use water again, then buffer, and then we treat this capillary with nitrogen. So this sequence can change, depends on your protein. So for some proteins, maybe it's necessary to use some ethanol, some detergent. So this is also something that you have to test on the beeline in the first test. And after testing this cleaning pipeline, then we can measure again the capillary, the empty capillary, just to be sure that it's empty, and then we can go on with measuring a lot of samples with different composition. So after measured and cleaning all the capillary, the last step, it's the data analysis. So as I mentioned before, we collect a lot of images, like thousands of images. So it's a really big data technique. And first thing is data reduction. The first step, we will crop the data around the region that we really have some scattering. And then we select this region of interest. And then from now we move only with the intensity from the region of interest. Then we apply the correlation function, and we have the correlation curve. And also in this pipeline, we can fit the data and then calculate the relaxation time that it's gonna be this data that we're gonna use to analyze. And usually, I forgot to mention it, on the beelines, all these pipelines are based on Python scripts, and then you have different pipelines depending on the beeline, and usually it's developed by the beeline team. And then for the last thing, most important, it's what information you get from this XPCS data. So using this KWW expression, we obtain the relaxation time. Then comparing the sample, we have information about how fast, how slow was the dynamics of condensates in different conditions. And another really important parameter, it's this gamma. Gamma, it's related with the shape of the exponential, and will give us information about the nature of the dynamics. So when you have single exponential, so gamma is equal to 1, it's characteristic when you have single relaxation time, like free diffusion, Brownian motion. And when you have like stretch exponential, gamma it's smaller than 1, we have a distribution of relaxation times. So for example, it's very common in complex liquids. And when you have gamma larger than 1, that's compressed exponential, it's characteristic when you have internal stress relaxation, so gels. So we used XPCS to characterize in vitro the dynamics of the PRP, and we had this information about relaxation times, and very important information to contribute to our work was this nature of the dynamics, because if you have a liquid state, then we can have like here Brownian motion, or gamma smaller than 1, that's complex liquid. But at the moment we have gamma higher than 1, it means that we have a gel phase. So combined with microscopy and in vitro and in vivo, we used XPCS in vitro to also have this information about the state of these condensates. Well, so with this webinar, I hope that it is clear for you that using the combination of these different microscopy and also XPCS, we can have this important information about the states of this condensate, and these really amazing results that Mariana showed in vivo in the cell, that it's really amazing. So we would like to thank all the people that contribute to this work. There's a lot of people, and here you have my and Mariana email. If you have any questions, please feel free to reach us. I'd like to thank you for your attention.
Thank you. Great, great. So I will read some questions here. I will start with the Q&A, because then I think you can answer live, and I was beginning to type some answers, but I think if you answer live, it will be better because everyone will be able to listen to you. So I have two questions from Katerina. One, is it possible to use cyper orange inside the cells? And the other one would be about the use of PRP signal at the contact between two cells. Is it known whether PRP forms homodimers? Okay, so I guess these ones are for Mariana and for myself.
So, thank you. It's a very nice question about cyper orange. Like, I've done some tests, but I didn't follow up. What I've really done is thioflavin S. So thioflavin S, it really works. We know that thioflavin T can also bind to nucleic acids. So thioflavin S doesn't bind. So for PRP, it really binds to thioflavin S, like some of the gel-like condensates. Cyper orange, I have to further optimize, so I'm not confident to say like, yeah, go ahead, you know. Regarding like homodimerization of PRP, yes, PRP can form dimers. We even see like in the SDS page, these dimers can be resistant to SDS, and it forms like oligomeric states very quickly, and we see that by DLS as well.
Thank you, Mariana. Regarding cyper orange, I think another drawback would be that it could bind other misfolded proteins
Yes.
in cells
Yeah,
as
the
well.
reason
Yeah.
why I
So
didn't
this,
go further as well, it's because we have the PRP-YFP construct, so it would overlap, would be very close. So that's why, like, I decided to use a dye that is spectrally separated. I also use AmiTracker 680. It's a really good probe, and some aggregates of PRP, sometimes they don't bind to thioflavin S, depending on mutations. So AmiTracker, as far as I know, it's more like broadly used for, it's being broadly used for PRP aggregates characterization.
thank you. So I think for the Q&A, the last one is from Sadala Surendra to Mariana as well. Can we also label RNA for the same purpose of LLPS along with an RNA binding protein? Thank you.
Yes, of course. Like, you can, if you know like the exact sequence of RNA that is the high affinity site, you can order these labeled RNAs with site 3 or site 5. We actually do that as well. So it's very important if you have like a multi-component condensates that you FRAP each component in the condensate because they can have different mobilities. So the protein can have full recovery, but the RNA sometimes doesn't recover, and vice versa. So in case of copper, because there aren't like commercially available fluorophores that are very specific for copper, we decided to only label PRP. But we are now pursuing like these fluorophores. We are collaborating with groups that develop them. So we can also probe, if copper is bound to a protein, and then this protein, because copper is never free at the cell. So we can also probe this protein bound to copper in the condensate where PRP is.
Thank you. So let's go for the chat. I will start with a question to Aline. So Aline Aline, the question here is from Bioprotocol from René. Besides proteins, what other types of biological material can you study with XPCS? Thank you.
Okay, thank you. Well, we use XPCS to study a lot of soft matter. So, for example, not in bio, but like colloids, polymers. And in biological materials, most of them are proteins, and we are also studying interaction of proteins with other materials. but main focus on the other material. For example, the diffusion of nanoparticles in protein media, and then we observe like aggregation or the formation of protein corona. So we are studying most like proteins, condensates, and also this interaction with different materials.
Thank you. There's a question from Carolina Jimenez, also regarding Cypro Orange. So why using Cypro Orange instead of Alexa Fluor or Eto dyes? I believe in relation for direct protein labeling. Yeah.
So we wanted to probe a little bit of the conformation of PRP in the condensates. So we use an extrinsic fluorophore. So when we conjugated PRP to Alexa, it was really to label the protein itself, but it's not informing, like at least in this scenario, about PRP folding. So it's really a challenge in the field to probe the conformation of proteins in the dense phase. So I think like the developments of new dyes will be very important to study that.
Thank you. There are some dyes that they are not new, but I was seeing that people are using to study condensates like Ekdan and also for the condensates that are close to membrane environments. So this could be also interesting for those who plan to follow up. Okay, another question. What is from Bat Ufak? What is the difference between round spherical droplets and oddly shaped aggregates? How to differentiate between two states and how to achieve these two states separately?
Okay, very good question. So one is the morphological assessment. But the morphological assessment, it can give you a clue about the material state, but it's really not probing that. So that's why it's so important to study the dynamics of these systems. So usually you have to combine both. So DIC is a very nice technique to see the morphology of the condensate, so you can compare. Usually like irregular aggregates are more solid-like, but it can also be not true. So you really have to test.
Good question. And there are two questions from Shun. One is why the condensate in vitro is not round. I guess he or she is asking about the wetting that was observable, and whether you use PEG for the XPCS measurements.
So yeah, so regarding the morphology, it really depends on the time the condensates are interacting with the cover slip, for example. So also the charge of the cover slip. So for PRP, we know that if we functionalize the cover slips with polylysine or PLL, it really interacts more with the cover slips, and over time it can wet the surface and the area of the surface is really occupied. And for example, hydrophobic glasses, we don't have a very remarkable wetting. Regarding like PEG, maybe Aline can also comment on that, but it's possible to use, but yeah, Aline.
Yes, we use the same buffer that Mariana showed for other experiments. So the idea in this case, it was using the same condition. But for sure, for example, if you use more PEG, you will slow down the dynamics. Could be the case when your dynamic is too fast and then you cannot study, and then it could be a strategy to slow down and then be able to get the dynamics. But in this case, it was the same composition.
Thank you, Aline. Well, and I'm seeing here people from all over the globe are attending the webinar. And I guess I read all the questions in the webinar chat, and they also put some questions of the previous Q&A. So are there any more issues and questions for the speakers? It was very pleasant to listen to your talk. Yeah, people are I think now you can access the chat, so you can read it. Yeah, yeah. And you can contact both of them or all of us as you have our email addresses. So it will be a pleasure to talk to you, and I guess for all of us, if someone is interested in doing some research in South America, in Brazil, and we are here, and we might have like open positions for PhD students or postdocs. Mhm. Katerina has a curiosity. You can type, Katerina. Wow. Very good question. Yes.
Yeah, so Katerina, actually, there are many biotechs, not many, but some that they have this vision of using like condensate modifying drugs that are called the CMODs. So maybe it is a possibility to really block this aberrant phase transition. But I do think like we have to really tackle like these in relevant cellular systems. So for example, I use hex cells because they are very amenable and very easily to manipulate. But this is lineage, right? This is like immortalized cells. So using like systems like IPCs or live neurons to probe the aberrant process is very important. But it is a very interesting view, especially because many of these proteins that form condensates, they are undruggable. So it's a really hot topic at the moment.
Yeah, this is the billion-dollar question. If someone can achieve that, yeah, for sure, future is guaranteed. Another question from Francisco Laurindo is, can you study two proteins at the same time? For instance, one inhibiting the phase separation of the other?
Yes, Francisco, that's a very good question. So, absolutely, absolutely, because one protein can form the scaffold of the condensate, and the other one can be a client and be recruited to that. Or you can have a different scenario, or both are drivers. But you can study that by turbidity assessments or microscopy. So, in case you incubate with an inhibitor, you will see that these droplets dissolve. And you can study that using microfluidity. So you can add the inhibitor, and in real time, you see the dissolutions of these condensates.
Great. Now I see. I think there are no more questions. Yeah, we reached our previous schedule of 1 hour webinar and Q&A. So if there are no more questions, I think I'll thank you, the speakers, the audience, Bioprotocol, for giving us the opportunity to be here and to share our work and the ideas that we have after the publication, also because this protocol is pretty much detailed. So I also recommend that everyone that still has doubt can take a deeper look in the protocol. So, and maybe some other questions will arise after it, because mostly for any of these assays, most of the questions will appear when you are doing the experiment yourself. So, and then new questions will come out, and then you can reach all of us. Okay, thank you.